Hello friends, I am Gaurang Makwana. I welcome you all to my another video tutorial of a Biotech guide. In this video lecture, we are going to discuss about a microarray, a tool for analyzing gene expression. So in this video lecture, we are going to discuss about basic principle behind this microarray technology, then what are the components are involved in microarray, then step-by-step procedure, application, and basic two types of a microarray technology. So this is our today's video topic. As usual, video is going on a very interesting level. Stay tuned with us and enjoy this video lecture. Let us talk about the principle. So the basic idea behind this microarray technique is hybridization. Hybridization, that means what? Formation of a H bond between two elements. Here first element is non-probe cDNA, which is immobilized on a microscopic slide, and another one is a unknown complementary sequence, which is our analyte will converted into a cDNA. So here a bond is formed basically, and bond is what? H bond. H bond is formed between these two elements, and a signal is generated via color formation. Here a signal is generated due to our analyte is labeled with a fluorescent. Okay, so the basic idea or basic concept behind this microarray technique is what? Hybridization. Now completely will brief in we are taking one example. Let us get a brief overview about a microarray. As we discussed in a principle, analyte is there and another complementary probe is there. So here analyte acts as these two cell and another probe, which is already bind with or hybridized with this microscopic plate or microarray plate. Okay, so for understanding the microarray tool or technology, we have taken two cell. First is a normal cell and another one is a cancer cell. Those two cells are further going for a culturing technique. Basically, after culturing, going for the mRNA extraction. After mRNA extraction, further going to be transferred for a cDNA formation via reverse transcription process. After cDNA formation, labeling is required. So normal cell should be labeled with a green fluorescent dye, another cancerous cell with a red fluorescent dye. After that, these two cell based this cDNA is further going to be transferred to this plate. Okay, as we discussed, this plate is already bound with a complementary probe. Okay, so what is happening? This plate is further going to for this scanner. Okay, so scanner has a three component: laser, computer, and a camera. Laser causes the hybrid bonds to a fluorescence. Then camera records the images produced when the laser scans the plate, and the computer allows us to immediately view our results, and it also stores our data. Here analysis is there. Okay, four colors you can observe: green, red, yellow, and a black. Green stands as a more healthy samples are hybridized healthy, that means a normal cell, and a disease, that means a cancer cell. Okay, so green indicates the more healthy samples are hybridized. Then red indicates the more disease samples are hybridized. Yellow stands both equal, healthy and disease both equal, and black indicates none of any hybridization place. Okay, areas where neither sample hybridized is to target DNA. So this is the simplest understanding behind this microarray technology. Now, what are the components are required for proceeding microarray technique? So basically 5 components are need to be proceed microarray technique. First one is a DNA chips, then target sample, fluorescent dye, probes, and a scanner. DNA chips is nothing but a microscopic slide. Basically, usually commercially available microscopic slide or DNA chips is used, made up of a glass, silicon, or a nylon. Each plate contains thousands of spots, and each spot contains a probe of a different gene. Then probe can be cDNA or synthetic oligonucleotide, such as a BAC, bacterial artificial chromosome. So these are the components Now let us discuss procedure. So basic 6 steps are there. First is a collection of a sample, then isolation of mRNA, then create and label the DNA, then after hybridization, microarray scanner, and at the end 6 analysis of a data. So we'll discuss one by one. First one is a collection of a sample. So here we use 2 sample. First one is a cancerous human skin tissue and another one is a healthy human skin tissue. These samples we have collected using cross section. Second, isolation of mRNA. Extract RNA from the sample using either a column or a solvent such as a phenol or a chloroform. Here we using column. After isolating RNA, need to isolate mRNA from the rRNA or a tRNA. We already know 3 types of RNA is there: mRNA, rRNA and a tRNA. But here we required mRNA for the synthesis of a cDNA. So we need to isolate mRNA from these 2. We already uploaded video regarding the types of RNA, link given in the description box, so you may visit out there. So for the mRNA isolation, we required one technique that is a column containing technique, where a column contains beads with a poly T tails. As you know the structure of mRNA, at the end poly A tails is there, which bind with a poly T tails. Okay, A bind with a T. So here poly A tail bind with a poly T tails. So that poly T tails is already present in the beads containing a column. So here we easily isolate the mRNA, but we are using this column containing technique. At the end we rinse the buffer, and the buffer disrupt the pH and resulted disrupting the hybrid beads. Okay, and that bead containing our mRNA. So here we isolate the mRNA via using column technique. Now formation of a cDNA and a labeling. So in third step, we add a labeling mix to the isolated mRNA. For the synthesis of cDNA, we already know that primer or a reverse transcriptase enzyme is required for the synthesis of a cDNA. Primer required for what? To initiate the process, and a reverse transcriptase enzyme which synthesize the cDNA. Then third, fluorescently dyed nucleotide which creating a complementary stain. Okay, so this kind of these 3 main elements are needed, and we add to the mRNA. Here another important concept is what? Fluorescently dyed nucleotides. So we labeled with them, we are using 2 different dye. First is cyanine 3, which is a green dye, we labeled with healthy cells, and another one cyanine 5, which is red in a color, and we labeled with labeled cancerous cell to this dye cyanine 5. Next, hybridization. So we apply created cDNA on a microarray plate or a DNA chip. When we are comparing the 2 samples, so we apply both samples on the plate. At that time, ssDNA, single stranded DNA of this cDNA which we created or generated will be bind to the cDNA already present on the plate, which is a known. Okay, so here a known probe which already present on the plate is bind with our analyte ssDNA. Next, scanning. So here microarray scanner is there, which has a laser, computer and a camera. All have a different function, like laser causes the hybrid bonds to a fluorescence, then camera records the images produced when laser scan the plate, and the computer allows to the immediately view our results, and it also stores our data. Now we'll see the result. Next, analyze the data or the result. So as you can see here, 4 kind of a color or signal is generated: green, red, yellow and a black. So green is the place where the healthy sample hybridized more than the disease sample with a target DNA. Then red stand the disease or cancerous sample hybridized more than the non-disease sample with a target DNA. Then yellow, both samples hybridized equally to the target DNA, and the black area where neither sample hybridized is to a target DNA. So this kind of a signal is generated. So by comparing the differences in a gene expression between these 2 kind of sample, we can understand more about the genetic kind of a disease. Now let us discuss the application. So these are the application. First is a gene expression monitoring. As we discussed in the example, measurement of absolute levels of expression for each represented gene by quantifying the amount of a target that hybridizes with with the array probes. So expression arrays is generated and is used for the gene expression monitoring or their further studies. Then second, diagnostic array. So diagnostic assays are playing an increasingly important role in a treatment of a disease. So that kind of arrays is also further used for the diagnosis. Then microbial detection and identification. So the most commonly used gene targets are 16S bacterial and 28S fungal and intergenic transcribed spacers ITS in rRNA genes. And microarray technology has been incorporated to compensate for the time-consuming sequencing and identification procedure. So microarray is also useful for the microbial detection and identification. Then detection of chromosomal abnormalities. So array-based comparative genomic hybridization, defining as ACGH, is a rapid method monitor for measure DNA copy number of changes like a detection or amplification. Then second, in detection of chromosomal abnormality is SNP based microarray. So SNP arrays contain oligonucleotide probes spotted systematically to detect the 2 alleles of a specific SNP locus. So this is also used in identify chromosomal abnormality. Then methylation pattern analysis. So changes in a methylation pattern usually generate alteration in a gene expression program. So is useful for analysis of a methylation pattern. Then genomic analysis. So microarray based genomic DNA profiling MGDP technologies are capable of detecting copy number of changes for the whole genome in a single assay. Then the protein microarray. So is an emerging technology that provides a versatile platform for the characterization of hundreds or thousands of a protein in a highly parallel and a high throughput way. So this kind of application are there for the microarray. Now we'll discuss types of a DNA microarray. So basic two types are there. First one is a cDNA based microarray and second one is a oligonucleotide arrays. So first cDNA based microarray. So as we discussed in this video, this is a type of a cDNA based microarray where chips is prepared called as a cDNA chips, probe DNA or cDNA microarray because the chips is prepared via using cDNA. Then cDNA is amplified using PCR. Okay, here one application is also there, PCR, where the amount of a cDNA is very low, is increasing via using a PCR. Then the immobilized on a solid support made up of a nylon filter or a glass slide 1 into 3 inches, and the probe DNA are loaded to a spin by capillary action, and then DNA is delivered mechanically or in a robotic manner. So this is the cDNA based microarray. Then second, oligonucleotide array, also defining as a gene chips. The main difference between this cDNA based microarray and oligonucleotide array is here short DNA oligonucleotide is used, and in DNA microarray cDNA is used. cDNA microarray cDNA is used, and here short DNA oligonucleotide are spotted on the array. Then small number of 20 to 25 oligomoles per genes are represented, and main feature that each gene is normally represented by more than one probe. So this is the main feature. And two kinds of technology is also used, Affymetrix and Agilent. Affymetrix is have application of photolithography, and Agilent have a inkjet printing technology. So these are the main types of a DNA microarray. So here we completed our video. I hope you enjoying very well and got it very well. Thanks for watching, and if you want to get more and more updates regarding like this, so join with us, subscribe to our channel, press the bell icon, never miss an update. Thank you so much friends.